Hi students, I welcome all of you to the general considerations of antimicrobial agents part 1. At the end of the teaching learning session, the student should be able to describe the importance of antimicrobial agents in therapeutics, define the terms antibiotic, antimicrobial agent and chemotherapy, describe the history of chemotherapy, classify the antimicrobial agents based on the different criteria. Now, whenever we have had a bout of pharyngitis or tonsillitis or even sinusitis, we were prescribed azithromycin and levofloxacin for 3 to 5 days. Similarly, ciprofloxacin is prescribed for bacterial diarrheas and dysentery, metronidazole for amoebic dysentery, ciprofloxacin and ceftriaxone for enteric fever, chloroquine for malaria. All these drugs are antimicrobial agents and they are also known as antibiotics. In fact, the terms antimicrobial agents and antibiotics are used synonymously. But they do have some difference we'll study in this lecture. Now, introduction to the antimicrobial agents. What is their importance in therapeutics? First of all, the greatest contribution of 20th century to therapeutics has been the antimicrobial agents. These agents have really changed the outlook of a physician as to the power a drug can have in therapeutics. Antimicrobial agents are different from the other drugs in that they can actually cure the infection, whereas if you take certain drugs like antihypertensives, anti-anginal agents, drugs used in asthma, drugs used in Parkinson's disease, Alzheimer's disease, they cannot cure the disease, but they can just reduce the burden of the disease by reducing the severity of the symptoms. Now, these agents are more important in developing countries probably because the infections are more here when compared to the developed countries, and these are the drugs which are most frequently used as well as misused, because of which there is a very dangerous situation called as antimicrobial resistance. Now, these drugs are special in the fact that they specifically inhibit or kill the infecting parasite with minimal or no effect on the recipient, that is the human being. And this type of action has been referred to as chemotherapy. This is in sharp contrast to the drugs used in other pathological conditions. For example, anti-asthmatics, they have to interfere with certain mechanisms within the human body to bring about their benefit. And the same applies to antihypertensives, anti-anginals and all those drugs. Now, why are these agents highly specific on the parasites? Why they are toxic only to the parasites, that is the invading microorganisms, and not to the human beings? This is because they act on a target site or a metabolic pathway which is unique for the microorganisms. Now, let us look at this example. Penicillin G is an antibiotic. It is an antimicrobial which is used for bacterial infections. This drug is highly specific against the invading bacteria. It is not toxic to the human cells because peptidoglycan is the target of action for penicillin G, and peptidoglycan is present only in the cell wall of the gram-positive bacteria. It is absent in human cells. So this forms the basis for the selectivity of antimicrobial agents in their toxic effects towards the microorganisms. Now, let us know some important definitions of terms like antibiotic, antimicrobial agents. So, antibiotic and antimicrobial agents, although they are used synonymously, they are quite different from each other. Antibiotic is a substance which is obtained from microorganisms and selectively suppresses the growth or kills other microorganisms at very low concentrations. For example, penicillin G is obtained from Penicillium notatum, which is a fungus, and penicillin G kills gram-positive bacteria. So it is obtained from a microorganism and it is used to kill other microorganisms. So that is an antibiotic. Now there are many synthetic drugs also which suppress the microorganisms, and to have them also under a single term, we have the term antimicrobial agents, which includes not only natural substances but also the synthetically derived drugs. And when we use the term antimicrobial agent, it includes synthetic and natural drugs which suppress microorganisms. Chemotherapy we have already discussed, that is when we use drugs to treat infections or even in the treatment of cancers, so that the drug has specific action on the invading microorganism or the cancerous cell with minimal or no effect on the host cell, then we call it as chemotherapy. This is in contrast to the term pharmacotherapy, which we use, like for example pharmacotherapy of myocardial infarction, pharmacotherapy of bronchial asthma. I cannot call it as chemotherapy of bronchial asthma. Okay, so chemotherapy is a term used when we use drugs for curing infections and treatment of cancer. The history of chemotherapy can be divided into three parts. That is the period of empirical use, Ehrlich's phase of dyes and organometallic compounds, and the modern era of chemotherapy. Now let us see what are the events in these different stages. Now empirical use means using substances in the treatment of certain disorders which does not have a scientific basis. It does not have a scientific background. There is no objective evidence that this substance is useful for the treatment of that particular disease, and the use of substances is based on experience. For example, use of moldy curd on boils. Chalmugra oil was used in the treatment of leprosy. Chenopodium was used for the treatment of intestinal worms. Mercury was used for the treatment of syphilis, and cinchona was used for the treatment of fevers. So when these compounds were used, there was no scientific evidence that these compounds are actually useful in the treatment of those disorders. That is the meaning of empirical use. Next was the phase from 1890 to 1935, Paul Ehrlich's phase of dyes and organometallic compounds. Now the later half of 19th century, it saw the demonstration of the phenomenon of antibiosis by Louis Pasteur. Louis Pasteur showed that an microorganism can kill another microorganism, and this he showed through the destruction of anthrax bacilli in the urine when it was exposed to the atmospheric bacteria. And it was also known at that time that microorganisms were the cause for many diseases. So taking this into account, Ehrlich he hypothesized that if coloring agents or dyes could selectively stain the microbial organisms, these coloring agents can also be selectively toxic. And he tried the various dyes in the form of methylene blue, tryptan red, etc. In fact, it was Ehrlich's laboratory which came out with the first drug for the treatment of syphilis, arsphenamine and neo-arsphenamine in 1806. So the first drug for syphilis was from Paul Ehrlich's laboratory, 1906 and 1909. And he also developed the arsenical compound etoxyl for the treatment of sleeping sickness. And Paul Ehrlich was the person who coined the term chemotherapy, because he used compounds whose structural chemistry was known. Most of the other compounds which were used the people were not knowing what was the chemical structure. And Paul Ehrlich was the person who showed that selective attenuation or selective killing of infecting parasite was practical. Paul Ehrlich was a German physician and a scientist in hematology, immunology, and chemotherapy. He is regarded as the father of chemotherapy. He coined the terms chemotherapy and receptor, and he was awarded the Nobel Prize for his work on immunity in 1908. Now comes the modern era of chemotherapy, which was ushered in by a scientist called Domack in 1935. Now Domack demonstrated the use of a sulfonamide dye called Prontosil in pyogenic bacterial infections. Now later on it was found that the active component of this dye was para-aminobenzene sulfonamide, and the coloring agent was not essential. In fact, the first sulfonamide to be marketed in 1938 was sulfapyridine. Gerhard Johannes Paul Domack was a German pathologist and bacteriologist. He is credited with the discovery of sulfonamidochrysosine, which is a sulfonamide dye, and it was made commercially available under the name Prontosil. And Domack was awarded the Nobel Prize in 1939 for his discovery of sulfonamides. Now, in the later part of 19th century, the phenomenon of antibiosis was demonstrated by Pasteur, who showed that the anthrax bacilli in the urine could be inhibited by the airborne bacteria. Keeping this as the background, Alexander Fleming in 1929, he was a microbiologist. He observed that a mold which was contaminating his culture plates was responsible for lysing or destroying the staphylococcal colonies in the culture plates. He was a botanist by profession, and he could identify the mold as Penicillium notatum, and he named the diffusible substance which was responsible for lysis of the staphylococcal colonies as penicillin. But unfortunately, in 1929, when he discovered penicillin, Alexander Fleming was not able to purify this compound. After 10 years after the discovery of penicillin in 1929, Chain and Flory, two scientists, took up the job of purifying penicillin, and they were successful in purifying this compound. This led to the clinical use of penicillin for the first time commercially in 1941. And that time the Second World War was going on. The importance of penicillin in treating the war wounds of the soldiers was understood then. So the commercial manufacture of penicillin gained a lot of importance during that time. Inspired by all these discoveries, Waxman, a scientist, and his colleagues undertook a systematic search for antimicrobial agents from soil actinomycetes, and they were successful in discovering streptomycin in 1944 from soil actinomycetes. And the soil actinomycetes, it proved to be a treasure house for antimicrobials. Many antimicrobials, tetracyclines, chloramphenicol, erythromycin, polyenes, were actually isolated from soil actinomycetes. So all these scientists who contributed to the discovery of the antimicrobials were awarded the Nobel Prize. Domack received the Nobel Prize in 1939 for the discovery of sulfonamides. Fleming, Chain, and Flory, they got the Nobel Prize for the discovery of penicillin G in 1945. Waxman received the Nobel Prize in 1952 for the discovery of streptomycin. Now, Alexander Fleming, who is credited with the discovery of penicillin in 1929, was a Scottish biologist, pharmacologist, and botanist. He shared the Nobel Prize with Flory and Chain. Selman Waxman is the discoverer of streptomycin in 1944. Now, how should we classify the antimicrobial agents? So these are the various criteria based on which we can classify the antimicrobial agents. One, based on the chemical structure, based on the mechanism of action, the type of organisms against which they are primarily active, the spectrum of activity based on the type of action, as well as the source from which we obtain the antimicrobial agents. So based on the chemical structure, this is how we classify the antimicrobial agents. We have sulfonamides like sulfadiazine, sulfones like dapsone, diaminopyridines, trimethoprim, pyrimethamine. We have ciprofloxacin, levofloxacin, which are fluoroquinolones. Nalidixic acid and norfloxacin are quinolones. Penicillins, cephalosporins, monobactams, carbapenems have a beta-lactam ring, hence they are called as beta-lactam antimicrobials. Oxytetracycline, doxycycline have 4 cyclic rings, hence they are called as tetracyclines. Gentamicin, amikacin are aminoglycosides. Linezolid is an oxazolidinone. Metronidazole, tinidazole are nitroimidazoles. Miconazole, clotrimazole, fluconazole are the azoles, which are antifungal agents. And since a large number of double bonded structures are present in amphotericin B and nystatin, they are called as polyenes. Now, different antimicrobials have different mechanism of action. They act on different structures within the bacterial or the microbial cell. So certain antimicrobials which inhibit cell wall synthesis like penicillins, cephalosporins, vancomycin. Similarly, there are certain antimicrobials which cause a leakage from the cell membranes. They are the polyenes like amphotericin B, polymyxins, bacitracin. Certain antimicrobials interfere with the protein synthesis. So we have tetracyclines, chloramphenicol, erythromycin and linezolid. Aminoglycosides like gentamicin, they cause a misreading of the mRNA code and hence they affect the permeability of the cell wall. Fluoroquinolones like ciprofloxacin interfere with the enzyme DNA gyrase and hence they interfere with DNA replication. The anti-tubercular drug rifampicin and anti-amoebic drug metronidazole, they interfere with the DNA function. The antiviral drugs like acyclovir, which is an anti-herpes drug, and zidovudin, which is used in the treatment of HIV and AIDS, they interfere with DNA synthesis. And certain antimicrobials like sulfonamides, sulfones, para-aminosalicylic acid, trimethoprim, pyrimethamine, they interfere with folic acid synthesis. And ethambutol also interferes with arabinogalactan synthesis in the mycobacterial cell wall. Now, based on the type of organisms against which the antimicrobials are primarily active, we have antibacterial agents which are used for bacterial infections, penicillins, aminoglycosides, macrolides. We have antimicrobials which are used in fungal infections, amphotericin B, itraconazole, fluconazole. Similarly, as we have seen, the anti-herpes agent, anti-HIV agent, and amantadine is an anti-influenza agent which are classified as antivirals. The anti-malarial chloroquine, the anti-amoebic metronidazole, they are classified as anti-protozoals. And drugs which are used for the worm manifestations, helminthic manifestations, mebendazole, albendazole, praziquantel. Antimicrobials have different spectrum of activity. Certain antimicrobials have an action only on a few bacteria, whereas certain antimicrobials, they act on many bacteria or many microorganisms. So based on that, we have narrow spectrum and broad spectrum antimicrobials. Penicillin G acts mainly on gram-positive organisms, so does erythromycin, hence they have a narrow spectrum. Tetracyclines and chloramphenicol, they act on both gram-positive, gram-negative bacteria, hence they are broad spectrum. Now there are certain antimicrobials which come in between. They have an intermediate spectrum of action, the extended spectrum penicillins, cephalosporins, fluoroquinolones. Now, antimicrobials, they can kill the microbes or they can interfere with the multiplication of the microbes. So we have primarily bacteriostatic antimicrobials like sulfonamides, tetracyclines, chloramphenicol, erythromycin, ethambutol, clindamycin and linezolid, whereas certain other antimicrobials, they have the ability to kill the bacteria or the microorganisms. So we have penicillins, aminoglycosides, cephalosporins, vancomycin, fluoroquinolones, metronidazole, rifampicin, isoniazid. These are all examples of antimicrobials which kill the microorganisms. Now, based on the source from where we obtain the antimicrobials, so we have penicillins, cephalosporins and rhizophalamin which are obtained from fungi. So as we have seen, penicillin G is obtained from Penicillium notatum. Similarly, polymyxin B, colistin, bacitracin, SGNM, they're obtained from bacteria. And as we have seen from the soil actinomycetes, we can get aminoglycosides, tetracyclines, chloramphenicol, macrolides and polyenes. So in this lecture, we have learnt about the importance of antimicrobial agents in therapeutics, the definitions of some important terms like antibiotic, antimicrobial agent and chemotherapy. We have learnt the history of chemotherapy and we have seen how we can classify the antimicrobial agents based on the various criteria. Thank you very much. Happy reading.